So our human sociality is grounded in the general sociality of mammals that some of you might have heard about last month. But there's something special about us primates that has a lot to do with our evolution, our biology. And you can see this here in this video of a group of Tonkian macaque monkeys, which literally and figuratively have their lives revolve around each other. And even though most of you will never have seen the species before, I think you can sort of appreciate the little drama that's happening here. There are about 5 individuals. You see the active facial displays. And it's thought that this is the beginning of our primate intelligence. The alternative hypothesis is shown here. That is the invention of the nose pick through the high dexterity of the primate hand. And it's thought that that's like another way that primates can explore and understand their environment by using their hands. But the key idea is that that the evolution of our primate intelligence really has to do with our sociality, because the most difficult thing for the mind to understand are the minds. And while we are trying to understand and predict the behaviors of others, they're trying to predict and understand ours. And so in this arms race, our primate intelligence is thought to have evolved. A lot of this has to do with the face. And I like this image here. If I show you a picture of a face, within a fraction of a second, you get all of these kinds of information out of this image. Identity, gender, age, race, species, familiarity, attractiveness, presumed trustworthiness, not actual trustworthiness, mood and attention. All of this you can get, and you will get, even if you don't want to. If you look at a face, you will form opinions about all of these different things. So there's something very important about our faces and the information that they convey. But we're also overestimating our ability to understand faces. We now know that in 70% of exonerations where genetic evidence made it clear that someone was falsely convicted, the reason or part of the reason for the false conviction was an error in eyewitness identification. And I would like to do like a little experiment with you on your ability as an eyewitness. I will show you 40 pictures in the next slide for a couple of seconds. It shows 40 pictures of faces, and afterwards I'll ask you how many different people were shown in these 40 different images. Okay, who thought that all of these individuals, all the pictures belong to one person? Tony Mofshin, okay. 2 different people, 3, 4, 5, 6 or more? No, only 6. Okay, so the majority says 6 or more. Some people have seen this before. It's actually 2 different, and I'm going to be outlining here one individual in green. And you can see some of the difficulties recognizing faces, even though these are all pictures with front views of faces, so there's no variation in head orientation, which is a major change. There's some changes in facial expression, some changes in lighting condition, few of age, maybe facial hair and so on and so forth, that makes it a little difficult to discriminate different faces. So let's walk through this. What is necessary for you to discriminate these faces? Well, the first process is face detection. So in this image here with very different lighting conditions, none of you will have difficulties to say that there are 3 faces there, where they are, even though they have different orientations, different distances, and so on and so forth. That process is called face detection, just knowing that a face is there and where it is. But for you to be a good eyewitness, you have to do more. If you see like multiple pictures like this one here, even though on a pixel by pixel basis, the front views of different identities are more similar to each other than different views of the same identity, perceptually, you're grouping these together and generate a representation something like this. We're not going to be able to discriminate different individuals across different viewing conditions. And this process is called face discrimination. So we'd like to know how this is happening in the brain, where is it happening in the brain, and so one tool that we've been using is to use functional magnetic resonance imaging, where we look at activity in the brain during the presentation of faces compared to activity in the brain during the presentation of non-face objects. And we found consistently there are 6 regions shown here in yellow and red at reproducible locations in the macaque monkey brain that respond more to faces than to non-face objects. So this really is a great advance for us in order to uncover the neural mechanisms by which you recognize faces, because our brains are very big, very complicated, and being able to reduce the area in which we have to look really makes this problem much more tractable. Okay, so within each of these regions that are yellow and red here, there are hundreds of thousands of neurons within each of them. And most of you will know that these cells look like this, really beautiful. They have branches like trees from which they receive their inputs. They have other processes, these so-called axons here, with which they can send information or activity to other cells. And if we can introduce a recording electrode near the cell body of these cells, we can actually listen to the activity by amplifying the signal and putting it on speakers. The cell is either inactive or active. It fires action potentials. We say when it's active, and we can put this on an audio monitor and listen to it. And I'll actually show you some experiments that we've done in the lab to illustrate this. So this is a way for us to monitor what kind of information a single cell has. And it is very difficult to imagine, unless you've taken a neuroscience class, that you could learn anything really meaningful from this, because these are very, very complicated cells wired into very, very complicated networks. And yet, in the 1960s, Charles Gross recorded from one part of the brain that lesion studies before it indicated were going to be important for the processing of objects, while he was presenting pictures of faces of monkeys or other biological objects, a scrambled version of the face, and so on and so forth. And he found cells like this one here would increase the discharge rate during the presentation of a face, but not during the presentation of a control object like a hand, or after other manipulations. You can see here this particular cell is responding both to monkey face, the human face. If you take out the eyes, the response is getting weaker. A pumpkin face also eliciting responses, but not a random collection of orientations, and so on and so forth. So you get the intuition, that the cell might be face selective and might really care not just about individual features, but about the configuration of the entire face. However, initially it was very difficult to document this. This is a paper from the early 1980s. The finding was made much before, when papers were written just describing what was done and was found. And many people in the field were really in disbelief that something meaningful like this, a cell that responded selectively to a face, would actually exist. So our strategy now was to combine fMRI. It allows us to localize these face areas and introduce a recording electrode. And I'm going to show you now one experiment that we did many years ago, where we recorded from a single cell inside of an fMRI-identified face region. You're going to hear the clicks whenever the cell is active, and you're going to see the stimuli that the monkey saw at the same time. Okay, so I hope that you can hear that every time there's a face shown, there's a response. The cell doesn't miss a single beat. When a face is shown, there's a response. It's not perfect in the sense that sometimes there are weaker responses when there's not a face there. But every time there's a face there, there's a response. And so we think that this is the reason why we have to see faces in the outside world. So I really like this picture of these peppers, who are unhappy because they've just been sliced in half. You know they're peppers, but you cannot not see them as faces, right? You have to see them as kicking and screaming pepper faces. Okay, so this is similar to face detection, right? The cell will respond when there's a face, and it's responding not at all, or weakly when there's no face. You can use it to detect whether the face is there. What about face discrimination? So if you have to discriminate between different identities, and we found cells that are very selective for this in a different face region over here that is connected to the one that is doing face detection. And to make it a little easier for you to see, these are the pictures that this particular cell is responding to, all from the same individual. Here the response is much sparser. There are many faces now going by that don't elicit a response. It's only the 8 different images from this one particular individual that elicit a response. So importantly, the subject has never seen these particular individuals in real life. So there's something about the physical properties of these particular images, despite the great variation across the different head orientations, that is eliciting a response, while other faces that might not look that different but have different physical properties are not eliciting a response. And so that's the main fundamental difference between these different face areas, which again, they're wired together into a network. All of these face areas have different properties. And so in two major transformations, you go from an early representation that will tell you if there's a face there or not, as good for face detection, to representation that's really called to facial identity. So that's the brief summary of this network here. So can we understand the properties of this network? And Richard Feynman famously said that what I cannot create, I do not understand. So we'd like to build computer models of these networks that capture the major properties in order to say, yes, we are able to understand what is going on in this network, therefore we can build it in silico. And so the first model that you might turn to is a deep convolutional network, which has been all the rage in computer vision and artificial intelligence for a long time, where you show an image and it's processed through several layers of processing, similar to how the network that I showed to you has several layers of processing. It's not very deep. It has maybe 3 or 4 layers of processing to it. And then if you train this network on lots and lots of examples, it's going to give you an output. It's going to tell you who is the person in this image. So this is one way that maybe this network could work. But Ilka Yildirim and Josh Tenenbaum and I thought that maybe the system works differently. In that, yes, it's using a deep network to process information from the face, but it's mapping the output not on a particular identity, but an internal 3-dimensional face model. It's sort of fitting the latent variables of this model. So if it's like a narrow face or a wide face, if the eyes are big or if they're small. So it's taking measurements of different parts of the face and then identifying these different parameters of this internal model. And only afterwards, the system would identify a particular individual. And if you have this model, it's, we call it inverse graphics network because it's kind of inverting the process by which you would generate the face that is here the input. So if you have this internal 3D model, you could actually generate images of faces at different orientations with different lighting conditions yourself. You could train yourself, but also could match now the input to this self-generated image to say, I'm able to understand what's coming in. So this is a more intelligent process of understanding the outside world because it allows you to recreate it and to use an internal model to understand the incoming information, not just passively map sort of like an output to the incoming image. So we have several lines of evidence why we think this is happening in the brain. One is that the neural data we record from the network fits the second model much better than the first. There are other arguments, but there's one that's very intuitive and I would like to show to you. And that is shown here. It's called the hollow face illusion. So what do you see here? It looks like Albert Einstein. It is a mask of Albert Einstein that seems to be looking at us. So we're now going to set it in rotation. It seems to be moving counterclockwise. Now something weird happened. So you might have realized that we looked into the hollow part of the face. Now it's looking at us. It's forward facing. And soon we're going to look into the hollow part again. And now something very weird happened, right? It's sort of forcing itself to be seen as forward facing, even though physically you're looking into the hollow part of it. I'll show it to you again. It's also the motion direction seems to be inverted. You're getting a full interpretation of what's shown here that's wrong. So soon I'm going to look into the hollow part again. It's popping out now and seems to be rotating the opposite direction. Isn't specific to Albert Einstein's face, but it's kind of more fun with his face. So this was what would happen if you had a 3-dimensional face model in your head. The only way for you to interpret this incoming information would be in terms of a forward-facing face, because that's the only way you could interpret it. Okay, so this is our understanding of this core face processing system. In essence, what it means is that when you see a picture of a face, you're actually getting more from it than is there physically in seeming violation of information theory, because you see it something like this. You're making a 3-dimensional interpretation. And this is a sign of intelligence because you're using a causal model to understand the incoming data. It's not that you're extracting something from massive amounts of data, but you're using an internal causal model to interpret this data. Okay. So I showed you this image before. I told you that there's a lot of information that's relevant for our social behaviors that you can gather from the face, like identity, gender, and so on and so forth. And the implicit assumption that I'm making here is that it's kind of a tableau of information from which you could pick, whatever you want, or maybe you have to pick everything. But it seems like we are active here, and the face is just presenting this information to us in this passive format. But far from this, faces are actually very efficient manipulators. So here's one example, a movie I've been showing for a long time. The reason being that this is a 3-day-old macaque monkey that's just been born with very little information about the outside world. And after some period of time, the experimenter is going to make some facial movements. And the little critter is going to be very interested in the face. Maybe he would be equally interested in bananas, but he's very interested in the face. And now that the experimenter is opening his mouth, it's getting very interested. And now it's mimicking this facial movement as best as it can. So I thought this is like really cool. Maybe there's something there at birth, for face perception. Maybe there's something there at birth for mimicking facial movements. And there might even be connections between, face processing sensory, visual circuits, and the output on the motor system. But then invariably I noticed that when I show this to the audience, there are a lot of like oohs and ahs in the audience going around, right, because he's so adorable. And I'm sure this happened to lots of you as well. You're completely defenseless against this, okay? So I'm showing you a 4-dimensional visual stimulus, but it's getting right into your emotional brain. It's going to manipulate you. You just have these, very strong feelings to the stimulus. It's not just the face. It's also, the fuzziness of the towel and, the movement patterns and all of it together, but you're going to have a very strong emotional response. So far from being like a passive tableau that you can pick from, the face is actually actively manipulating you. Okay, also works for baby faces. So here's a little demo I have for you for another way that the faces can manipulate you. So I have these glasses here. These are still my real eyes. But you have a very strong sense that I'm looking this direction. Am I looking this direction or that direction? This direction. Okay, very good. So your attention is going to be drawn that direction. You might think something's happening at that door there. Actually, once I did this, someone came through that door. Probably not going to happen now. So your attention is drawn in this direction that I'm looking at. And then after some time, it comes back to my face because you realize there's nothing happening there. But still, I seem to be looking there. So there must be something interesting there, right? And so you're going back and forth, back and forth. It becomes annoying over time, so to take off the glasses. But so this is how gaze is manipulating your attention system, right? So you have to look into this direction. And so it's been used on a British comedy show. And then lastly, you see this picture here. Something gets into your memory system, right? It's not just that you see a face, but you recognize a person that you don't personally know, but as a famous person that you know of. So there's a strong connection between face perception systems and memory systems. In fact, it's so strong that often we are confounding these two. Okay, and this gets us back to our little experiment that we did earlier about difficulties in eyewitness identification, discrimination. The people who ran the studies, by the way, 2 of these authors are the 2 individuals shown these images here. They ran a very interesting control. So they used the pictures of Dutch celebrities, of 2 Dutch celebrities, to make these 40 images, and then asked people again, like, how many different individuals do you see? And they gave it to British subjects and to Dutch subjects. British subjects on average, maybe like you said on average, there are like 7 to 8 different individuals in here. Dutch subjects, without a single mistake, all said there are exactly 2 individuals in there. So your personal familiarity with faces fundamentally changes your ability to discriminate them. You're not going to make the mistakes that you're doing with unfamiliar faces. And so part of the difficulty for eyewitnesses is that we're overestimating our ability to discriminate the faces of people that we don't know in person. So if you've seen The Godfather, you might remember the names, so you can now discriminate them and really recognize the different people in here. So we're very interested, where is this happening in the brain? Is it happening in the circuit that I showed you before with the 6 different face areas all wired together, or is it happening elsewhere? And this is really the work of from a grad student, one of her grad students, Sophia Landi. And so she looked again with fMRI, the different parts of the brain that are more active, but now to familiar faces than to familiar objects. It wasn't Ruth Bader Ginsburg, and as shown here by the example, but this is sort of the logic of the experiment, right? You're comparing now familiar faces with familiar objects. So what she found was the same set of face errors that we've seen for many, many years before, shown here. But in addition, she also found two errors that we had not seen before, one in perirhinal cortex and one in the temporal pole region. So perirhinal cortex was a region that we had hoped we might find here because previous experiments had shown that there are cells in there that can learn associations between different stimuli. So it's a region that's particularly plastic and known to be linked to visual processing. The temporal pole region, probably I should have been able to guess, but I didn't guess beforehand. There was a big surprise. Why should I have guessed it? Because people with lesions to the temporal pole have a person recognition deficit. It's like they forgot about people. It's not that they can't recognize faces, but they forgot about people. So it seems to be a region very much involved in the processing of relating faces to knowledge about people. Okay, so this is suggestive that maybe these regions that we haven't seen before now come up for this contrast, but of course it doesn't prove that these regions are really involved in the processing of familiar faces. And so we ran another experiment that again I'm going to do as an experiment with you guys. So in this experiment we are starting with highly blurred images, and probably those of you sitting in the back can recognize that these are face stimuli that are highly blurred, but they're so heavily blurred that could also be a tomato or a potato or and so we're going to be very, very slowly unblurring these images over the course of a half a minute. So when you recognize something in the image that you're familiar with, shout it out. Yeah, amazing, right? It's still like highly, highly blurred, but for people that we're very familiar with, we don't need the detailed information of individual face parts. It's sort of like the gist of the face that is sufficient for us to recognize them. And so by now, probably everyone has recognized Obama. Okay, so this goes on for a half a minute. Okay, another one. I heard that, yes, but it's not MLK. Okay, so this experiment works really well because you're not supposed to recognize this person. This is a half-brother of Obama's that most of you will never have seen or heard of. And so this is the logic of this experiment, right? If you have a generic face processing system, as I'm revealing more and more information, the area might become more and more active, maybe in a linear fashion. But if there's a recognition process going on where, irrespective of the fact that I'm presenting more and more information, you don't recognize the person until at a certain point in time, you have this aha moment, this eureka moment, that you now recognize this person, there might be this nonlinear response. And so this was the idea of the experiment, that this would be like a signature of a person or a face familiarity recognition unit. And so when we look at areas in the generic face processing area, there's a slight advantage of familiar faces over unfamiliar ones, but we see this sort of linear increase for generic face processing. And then these two regions that Sophia discovered, we see this nonlinearity, in particular in the temporal pole area, but also in the perirhinal area, and it's selectively only for the familiar faces and not for the non-familiar faces.
What were the subjects asked to do?
Nothing, just to look at it. Yeah, so we don't know if they actually did the recognition. All we know is that these areas showed this nonlinearity. Okay, and so if we go in and record from this region, we find some very interesting cells, like this one here. So here what you're seeing from top to bottom are different stimulus conditions. From left to right is time. And in a color-coded fashion, where red is indicating response enhancement, you can see that there's one image and one image only. It's the image of like one familiar individual for the subject animal that this cell is responding to. So this cell is very selective to like one personally familiar individual. So those findings together convince us with others that there is something about this temporal pole region here that is there for processing information about familiar individuals. Okay, so what have we learned? In 1986, two face recognition researchers, Vicky Bruce and Andy Young, proposed a model of face processing, which they called a functional model of face processing, where they suggested that if there's an image coming in, it's being processed here. through first the view-centered description, and I didn't mention this to you. The first area that I showed you that I mentioned is involved in face detection is actually very, very sensitive to head orientation. It's then transformed to the representation that is invariant to changes in expression or different head orientations. And then it goes to face recognition units. And so that's exactly what we found. It's pretty amazing. I had an exchange with Andy over it, like how was it possible for them to predict this so well? And he was very modest in saying, well, we didn't actually predict it so well. But this core face processing system is pretty much doing exactly this. And then this area TP is doing pretty much exactly this. I didn't show to you, it's not responding to voices, other things that are selected for a particular individual. It's also not responding to bodies. It's really just about the face. And it's not yet a person identity node as shown here. So the thing I struggle most with is like, how were they able to predict that there should be a face recognition system? Why shouldn't it go directly to a person recognition system? But that is their proposal. So what we've learned is that there's something about this temporal pole region that makes it very interesting and very selective. And for us, this is very interesting because this region is primate specific. So humans have it, other primates have it, mice don't have it. There's something primate specific about this particular part of the brain. Here it is shown again the recording from the monkey brain, and in red is the region in your brain. It would be somewhere here. There's not much known about this region. In fact, the most important review article calls it the enigmatic temporal pole. So we know some of the things, but we really can't put this together. And for us, it was this finding really was a reason to look again in humans, where we know something about face processing from many years of fMRI. But we didn't know where the area was going to be located that was processing information about familiar individuals. And so Bandino was an amazing postdoc in the lab. He took this on. He actually had to reprogram our scan sequence in order to get a signal in this part of the temporal pole. It's very difficult to image in order to get a signal from this region. And so he parcellated this part of the temporal lobe. So there's the temporal pole region here, anterior SDS, anterior IT, and the perirhinal region that we also identified in the macaque monkeys. And he found that, yes, this region is responding selectively more to familiar faces than to unfamiliar faces or to any other control stimuli like objects, unfamiliar scenes, or familiar scenes that we might show. So the functional model from Bruce and Young would predict that there's further processing from face recognition to person identity, maybe name retrieval, and then there's interaction with cognitive systems because you can think about faces, and so there has to be like some connection. But Ben was really interested to try to figure out whether this temporal pole region of the human brain was only selective to faces as predicted by this model, or if it would have a more general involvement in social information processing. And so for this, we had a very painstaking process with our IRB that we could recruit individual subjects who would come back multiple times to the MR scanner, who we could then contact friends and family members from, so that we could get pictures of individuals that they are personally familiar with, that we could also get information about things that have happened in their past, and information about these other people. Everything was kept confidential, so no concerns there. So why did we do this? Because now we could compare activations in very different tasks. So we have on the left-hand side the visual task that I showed you before, where there are faces, familiar, unfamiliar, where there are scenes, familiar, unfamiliar ones, from the person's individual subject's past. This is how we found this region in the first place. But we have other tasks where in the semantic and the episodic condition, there are no visual stimuli, at least no faces or no scenes shown. These tasks went like this. So semantic information is information about individuals that they know. Is the person shy? Is the object rough? Is this place spacious? Things like this. So information that they would have about places and people that they know from their past. In the in the episodic condition, we asked them to imagine a scenario in the future where they would be interacting with this person and where they would talk about a particular subject, for example, and how this person might react. So absolutely no visual information there, about faces or places, but a lot of information from semantic information through this description of these episodic scenes. And then we have other controls like a dynamic visual condition where we're showing faces and objects in motion. This is another independent control. And then we have a so-called theory of mind localizer that allows us to assess whether the subjects might form representations of internal states attributed to others, like what others might be thinking, for example. This is like a standard localizer to localize parts of the brain involved in high-level social cognition. Okay, so what does he find? For the left and the right temporal pole region, he finds again this difference between the familiar and the non-familiar phase stimuli. But he also finds activation selectively in the semantic and the episodic condition, again, where there's no face image shown, just by imagining or reading about familiar individuals. And this, again, very specific about familiar individuals, doesn't happen for objects or places. Then there's a response to moving faces that is sizable, and there's even a response in the theory of mind condition where they have to form opinions about, or make mental state attributions to others. This does not happen in periviral cortex. There's a small effect of familiarity here. It's also responding to faces in motion, but periviral cortex is not engaged by these more cognitive memory and imaginative processes. And also general face processing area like the occipital face area, which only shows a very small advantage for familiar faces, also responding to moving faces at the same level. It doesn't show the selectivity for these cognitive conditions. So this is taking advantage of the fact that with human subjects, you can easily instruct them to do things that would be difficult to do with animals. The next thing that we wondered about is whether this temporal pole region might be coupled to parts of the brain that are known to be deeply involved in high-level social information processing. And they are. So this is a connectivity study of the temporal pole region, and it shows the strongest activations in posterior parietal cortex, medial prefrontal cortex, and the temporal parietal junctions. These are the three regions typically found in these theory of mind tasks. So when you are thinking about the thoughts of others and making inferences about what people might be doing based on their internal states, use these areas. Periviral cortex is not so much connected to these, but more to other parts of the visual system. So this is shown here. The temporal pole region is really the one that's interacting the most with the social brain. Periviral cortex region, again, like the face selective part of it, is interacting more with the visual system. And so we're proposing like a two-pathway model where the temporal pole region is really a link between face processing regions and a social information processing network, while the periviral cortex is more an extension of the visual system into memory. And so because of these connections, we're now also curious, how is information in these different tasks processed in these different regions. And this is, again, the response profile of the temporal pole region. And this is the region, medial prefrontal cortex. And you can see the profiles are almost identical to each other. So in deviation from this functional model from Bruce and Young, we find that the temporal pole region is definitely part of a larger system for social cognition and might already contain processes that you would call social cognitive processes. Okay, so I showed you that our very complex brains can be studied in a systematic fashion. Behind the complex brains is a complex connectivity structure. I showed you how we can use fMRI and single-cell recordings to dissect these circuits. I showed you different functional specializations in a particular subnetwork of the social brain that is processing visual information, and how we can capture some of these processes in computational models. And lastly, I was showing how the region that is implied here, that is relating an internal face representation to knowledge about particular individuals, is actually part of the larger scale social brain. So thinking about the social brain and its organization, I would like to conclude. Actually, I'm very fast when I talk. I'm not sure what I was thinking. But three final remarks. So think about the social brain. You see these activations in very different parts of your brain, right? And so there are connections behind it, white matter tracks that connect between these different brain regions. And so there must also be cells that are sending information from one part of the brain to the other, so-called projection neurons. And so we're very interested in these particular cells, the genetic profile, and their functional properties. It's in part, I'm at this place here that's very much devoted to the understanding of autism, that these cells that are projecting from the temporal to the frontal lobe are particularly vulnerable developmentally in autism. And so this is one of the reasons why we're particularly interested in this brain region. Second, I showed you different functional specializations that occur in the brain, but you might wonder, like, why do they occur where they occur? And we also wonder about this, and we particularly wonder about the relationship to possible genetic specializations. So we're part of a consortium of the Brain Initiative, and our component is to relate the functional specializations for faces and for other processes to gene expression specializations that might explain why these functional differences exist, or that might explain why particular processes are particularly vulnerable in particular disorders. And I mentioned this briefly, or you might have seen this briefly, is that we see specializations even within the social brain regions. So in different parts that are traditionally assigned to the social brain, we also see specializations for spatial processes. And so this section of functional specificity has to be done with a very high spatial resolution for us to be able to extract the genetic principles. And then finally, I would like to solve for the mystery man in my initial picture. Who knows who this is? Yeah, it's Martin Buber, great. So normally no one knows Martin Buber, who in 1923 published this book, Ich und Du, I and Thou, in which he lays out his understanding. It's an epistemological proposal that there are two ways in which we can understand the world. One is I and it, so I and the world, like we as scientists understand the world. And the other one is the interactive or dialogical one between I and you, which I think is sort of the fundamental to thinking about the social brain. So again, I'm very early. I don't know what I was thinking. I'm sorry for that. So I'd like to thank my funding sources, in particular the Simon's collaboration on the global brain. Tony Mofschen is here. This has been a great support of funding for us, and also like a reason for me to come here. So thank you very much for your attention.